Chapter 9: Microbial Growth. Biofilms, seen as slimy layers on pond surfaces or slippery rocks, are organized ecosystems of microorganisms, including various species of bacteria, fungi, and algae, embedded in a protective matrix. Although once thought to be random assemblies of cells, advancements in visualization and biochemical methods have highlighted their complex interactions through cell signaling and coordinated responses. Biofilms provide a safe environment for microorganisms under harsh conditions and are clinically significant, forming medical devices and contributing to health-acquired infections such as plaque on teeth, lung infections in cystic fibrosis patients, and cardiac issues in endocarditis patients. Their resistance to cleaning up and sterilization poses challenge for treatment, necessitating new research methods to study their adhesion properties and growth in natural environments. The generation time for growth in microorganisms is defined by the process of binary fission, which involves simple cell division and follows typical phases observed in growth curves. Various laboratory methods are utilized to determine both viable and total cell counts in populations undergoing exponential growth. Additionally, alternative forms of cell division such as budding and fragmentation are important to recognize. Biofilms, which are communities of microorganisms, present health risks that must be addressed, and their formation and characteristics play a significant role in these risks. Quorum sensing is a crucial mechanism for cell-to-cell communication, allowing coordination of cellular activities. The bacterial cell cycle is marked by asexual reproduction, where DNA is replicated and divided into two daughter cells, primarily involving single circular chromosome, although exceptions like Borrelia burgdorferi exist, which has a linear chromosome. Most bacterial and archaeal cells reproduce by binary fission. Some bacteria reproduce by forming a bud or multiple fission. All must replicate and segregate the genome prior to division. Before the cell divides, it grows and increases in number of its cellular components. The replication of DNA begins at a specific spot on the circular chromosome known as the origin of replication, where the chromosome is attached to the inner cell membrane. This replication proceeds in opposite directions along the chromosomes until it reaches the terminus. The cell cycle is the entire series of events that occurs from formation of a new cell until its next division. It consists of three main phases. First, the B period, which is a period of growth following the birth of the cell. Second, the C period, during which the chromosome replication and partitioning take place. And finally, the D period, known as the cytokinesis, where a septum is formed, leading to the creation of daughter cells. The cell cycle starts with reaching the initiation mass, where a cell shows the origin of replication, chromosome terminus, and the bacterium. The second step is the initiation of replication, where a cell shows replisome. The origins separate, the cell elongates as the chromosome replicates, and partitioning continues. The third step is the formation of the septum after the formation of the Z ring. Chromosomes separate as separation continues. At this point, the cells divides, one leading to the initiation mass stage. In the intricate world of bacterial cell division, the PAR-ABS system stands out as a critical player in ensuring the accurate partitioning of chromosomes during cell replication. This system comprises three key components: PAR-A, PAR-B, and the PAR-S site on the chromosome. The process begins with the bacterial chromosome is replicated, resulting in two identical copies that must be segregated into daughter cells. At the heart of the mechanism is the PAR-S site, a specific DNA sequence located near the origin of replication. Once replication occurs, PAR-B proteins bind to these PAR-S sites, forming a large complex. This binding is crucial as it marks the newly replicated chromosome for partitioning. PAR-A and ATPase then interacts with the PAR-B and PAR-S complex. PAR A undergoes a conformational change upon ATP binding, which enables it to bind to the cell membrane and create a dynamic gradient of PAR A concentration within the cell. As the PAR A gradient develops, it facilitates the movement of PAR B and the PAR S complex towards the pole of the cell. The underlying mechanism is akin to the tug of war, where PAR A filaments pull the chromosome copies apart. This action ensures that each daughter cell receives one complete copy of the chromosome. Through this elegant system, bacteria can maintain genomic integrity and stability, even in the face of various environmental stresses. The PAR-ABS system exemplifies the remarkable adaptation of prokaryotic organisms, highlighting the complexity of their cellular processes despite their relatively simple structures. The center of the enlarged cell constricts until two cells are formed, with each offspring receiving a complete copy of the parental genome and a division of cytoplasm, or cytokinesis. This process of cytokinesis and cell division is directed by a protein called FTSZ or FITZ. FITZ assembles into a Z-ring on the cytoplasmic membrane. The Z-ring is anchored by FITZ-binding proteins and defines the division plane between the two daughter cells. Additional proteins needed for the cell division are added to the Z-ring to create a structure known as divisome. The divisome activates to synthesize a peptidoglycan cell wall and construct a septum that separates the two daughter cells. The daughter cells are divided by the division septum, where all of the cell's outer layers, the cell wall and outer membranes, if present, must be remodeled to finalize division. For example, specific enzymes break the bonds between the monomers in peptidoglycan, allowing for addition of new subunits along the division septum. The bacterial divisome is a crucial cellular structure responsible for facilitating cell division in prokaryotes. Composed of a complex of proteins, the divisome orchestrates the process of binary fission, whereby a single bacterial cell divides into two daughter cells. The formation of the divisome begins by the recruitment of the protein FITZ, which polymerizes to form a ring structure at the future division site known as the Z-ring. The ring serves as a scaffold for other essential protein, including a FITZ-A or FTSA, which anchors the Z-ring to the inner membrane, and other division proteins like FTSI, FTSK, and FTSQ, which play roles in the peptidoglycan synthesis and chromosome segregation. As the divisome matures, it coordinates the synthesis of new cell wall material at the septum, allowing the cell envelope to expand and ultimately leading to the separation of the daughter cells. The process is tightly regulated to ensure accurate division and cell size maintenance. Additionally, environmental signals and cell cycle checkpoints influence divisome assembly and function, emphasizing its role in bacterial growth and reproduction. In eukaryotic organisms, generation time refers to the duration between the same points in the life cycle of two successive generations. For instance, the typical generation time of a human is approximately 25 years. This definition is less applicable to bacteria, which can reproduce quickly or remain dormant for thousands of years. In prokaryotes, bacteria, and archaea, generation time is also known as doubling time, defined as the time it takes for a population to double through one round of binary fission. Doubling time for bacteria can vary significantly. For example, Escherichia coli can double in as little as 20 minutes under optimal laboratory conditions, while the same species may take several days to double in particularly harsh environment. Most pathogens like E. coli grow rapidly. However, there are exceptions. For instance, Mycobacterium tuberculosis, the agent of tuberculosis, has a generation time of 15 to 20 hours. In contrast, Microbacterium leprae, which is responsible for the Hansen's disease or leprosy, grows much more slowly with a doubling time of 14 days. Microorganisms grown in a closed culture, also known as batch culture, do not have nutrients added, and most waste is not removed. They follow a reproducible growth pattern known as the growth curve. An example of a batch culture in the nature is a pond where a small number of cells grow in a closed environment. The culture density is defined as the number of cells per unit volume, and in a closed environment, it also measures the number of cells in a population. While infections in the body do not always follow the growth curve, correlation can exist depending on the site and type of infection. When the number of live cells is plotted against time, distinct phases can be observed in the curve. The lag phase. The beginning of the growth curve shows a small number of cells known as the inoculum that are introduced to a fresh culture medium, which is a nutritional broth that supports growth. This initial phase of the growth curve is called the lag phase. During this phase, cells are preparing for the next stage of growth. Although the number of cells does not change during the lag phase, the cells grow larger and are metabolically active, synthesizing proteins necessary for the growth within the medium. If any cells are damaged or shocked during the transfer to the new medium, repair occurs during the lag phase. The length of the lag phase is influenced by various factors, including the species and genetic makeup of the cells, the composition of the medium, and the size of the original culture. In the logarithmic or log growth phase, also known as exponential growth phase, the cells actively divide through binary fission, resulting in an exponential increase in their number. For any specific bacterial species, the generation time under particular growth conditions such as nutrients, temperature, pH is genetically determined. This generation time is referred to as the intrinsic growth rate. During the log phase, the relationship between the time and the number of cells is exponential rather than linear. However, the growth curve is often represented on a semi-logarithmic graph, which gives the appearance of a linear relationship. Stationary phase. The stationary phase occurs as cell growth slows down due to various factors, including the accumulation of waste products and gradual depletion of nutrients and oxygen. These unfavorable conditions led to a plateau in total number of live cells, where the rate of new cells created by division equals the number of cells dying. During this phase, the culture density remains constant, and the carrying capacity varies depending on the organism type and specific condition, but it remains stable for a given organism under consistent circumstances. In the stationary phase, cells transition to a survival mode of metabolism, resulting in a reduced synthesis of essential components like peptidoglycan, proteins, and nucleic acid. This reduction makes stationary cultures less vulnerable to antibiotics targeting these processes. Additionally, bacteria capable of sporulation may enter this process during the stationary phase. The secondary metabolites, including antibiotics, are produced, and pathogenic bacteria may express virulence factors that enhance their ability to survive and spread within the host. For instance, in Staphylococcus aureus, quorum sensing triggers the production of enzymes that facilitates tissue breakdown, allowing bacteria to access nutrient-rich environments. The death phase. As a culture medium gathers toxic waste and nutrients become depleted, the number of dying cells increase significantly. Eventually, the count of dying cells surpasses that of dividing cells, resulting in an exponential decline in the overall cell population. This phase is aptly named the death phase, sometimes referred to as the decline phase. Many cells lyse, releasing nutrients back into the medium, which helps the surviving cells to maintain viability to form endospores. A few cells, known as persisters, exhibit slow metabolic rate. Persister cells are of medical significance as they are linked to certain chronic infections such as tuberculosis, which do not respond to antibiotic treatment. Long-term stationary phase. Bacterial population continually evolves, process marked by successive waves of genetically distinct variants, and natural selection occurs within a single culture. During the log phase, the relationship between time and the number of cells is exponential rather than linear. However, the growth curve is often represented on a semi-logarithmic graph, which gives the appearance of a linear relationship. The growth pattern illustrated in slide number 13 occurs in a closed environment where nutrients are not added and waste along with dead cells is not removed. However, in many situations, it is beneficial to keep cells in a logarithmic phase of growth. An example of this is found in industries that harvest microbial products. A chemostat is a bioreactor that maintains a microbial culture in a controlled environment, allowing for continuous cultivation of microorganisms. This device is vital in microbiology and biotechnology for studying cell growth, metabolism, and production of biological products. The term chemostat derives from two words, chemical and station, emphasizing the controlled chemical environment it provides. In a chemostat, nutrients are continuously supplied while waste products and excess cells are removed at a constant rate. This process allows the culture to reach a steady state where the growth rate of microorganisms equals the dilution rate. The chemostat can be operated in either batch or continuous mode, with the continuous mode being more common for long-term studies. One key advantage of using chemostat is the ability to maintain specific growth conditions such as pH, temperature, and nutritional concentration. Researchers can manipulate these parameters to observe their effects on microbial growth and product formation. Chemostats are widely used in various applications, including the production of antibiotics, enzymes, biofuels, as well as ecological studies to understand microbial interaction in natural environments. Overall, chemostats are essential tools in advancing our understanding of microbial physiology and industrial microbiology. Estimating the number of bacterial cells in a sample, known as a bacterial count, is a common task for microbiologists. The bacterial count in a clinical sample indicates the extent of an infection. Quality control for drinking water, food, medication, and even cosmetics relies on estimates of bacterial counts to detect contamination and prevent disease spread. Two primary approaches are used to measure cell numbers. Direct method involves counting cells, while indirect methods depends on measuring cell presence or activity without counting individual cells. Both direct and indirect methods have their own advantages and disadvantages for specific applications. Direct cell count is a method used to estimate the number of organisms in a liquid culture or on a plate by counting the cells directly. A common technique for this is the direct microscopic cell count, which utilizes a specialized slide known as a Petroff-Hauser chamber. This chamber is similar to a hemocytometer and features an etched grid of squares of varying sizes. A known volume of culture is placed in the chamber, and the cells are counted under a light microscope. By counting cells in several squares and calculating the average, one can estimate the concentration of cells in the original sample, while also accounting for any dilutions made prior to counting. Petroff-Hauser chamber offers several advantages, including ease of use, speed, and low cost. However, it is less effective for dilute cultures as there may not be sufficient number of cells to count accurately. To obtain reliable measurement, it is important to count cells in multiple squares and take an average. Overall, while direct cell count is a straightforward method, careful consideration must be given to the concentration of the sample to ensure accurate results. Using a counting chamber may not provide an accurate count of live cells, since it can be difficult to differentiate between live cells, dead cells, and debris of similar sizes under a microscope. However, recent advancement in fluorescence staining technique allows for distinction between viable and dead bacteria. These viable stains or live stains attach to nucleic acid, but the primary and the secondary stains have different abilities to penetrate the cytoplasmic membrane. The primary stain, which emits green fluorescence, can pass through intact cytoplasmic membrane, staining both live and dead cells. In contrast, the secondary stain, which fluoresces red, can only stain cells if the cytoplasmic membrane is significantly damaged. Consequently, live cells emit a green fluorescence because they only uptake the green stain, while dead cells appear red since the red stain replaces the green stain on their nucleic acid. Another method employs an electrical cell counting device known as a Coulter counter to detect and count variations in electrical resistance within a saline solution. A glass tube featuring a small opening is immersed in an electrolyte solution. A first electrode is positioned inside the glass tube while a second electrode is situated outside of it. As cells are drawn through the small aperture in the glass tube, they momentarily alter the resistance measured between the two electrodes, and this charge is recorded by an electronic sensor as shown in the illustration. Each variations in resistance corresponds to a single cell. The technique is both rapid and accurate across a range of concentrations. However, if the cell culture is overly concentrated, multiple cells may pass through the aperture simultaneously, potentially distorting the result. Additionally, this method does not distinguish between live and dead cells. The viable plate count, often referred to as a plate count, measures the number of viable or live cells. This method is based on the principle that viable cells can replicate and form visible colonies when incubated under appropriate conditions. The results are typically reported as colony-forming units per milliliter or CFU per ml. Instead of cells per milliliter, as the multiple cells may land in the same area, leading to a single colony. Additionally, bacterial samples that grow in clusters or chains are challenging to disperse, meaning a single colony can represent several cells. Some cells are classified as viable but non-culturable, which means that they will not form colonies on solid media. These factors contribute to the viable plate count being considered a conservative estimate of the actual number of the cells. Nevertheless, the method remains useful for estimating live bacterial populations. Microbiologists usually count plates that contain between 30 and 300 colonies. Samples with fewer than 30 colonies do not yield statistically reliable numbers, while plates with more than 300 colonies complicate the accurate counting of individual colonies. By focusing on this optimal range, the likelihood of multiple bacterial cells forming a single colony is minimized. Consequently, the calculated CFU provides a closer approximation of the actual number of live bacteria in the sample. There are two primary techniques for inoculating plates for viable counts: the pour plate method and the spread plate method. Although the final inoculation process differs between these two techniques, they both begin with a serial dilution of the culture to ensure accurate counting of viable cells. This slide demonstrates the steps in the pour plate technique and is self-explanatory. Two photographs, 1A, 1B, in the previous slides also show this method. This slide demonstrates the steps in the spread plate technique and is also self-explanatory. The slide number 20, photograph 1C, shows the method. The serial dilution of a culture is a crucial initial step before using either the pour plate or spread plate method. The main objective of the serial dilution is to achieve plates with colony-forming units in the range of 30 to 300. This process typically involves several dilutions in multiples of 10 to simplify calculations. The number of serial dilutions is determined by a preliminary estimate of the culture density. A visual representation is available to illustrate the serial dilution method. In this method, a fixed volume of original culture, say 1 ml, is added to and thoroughly mixed with the first dilution tube solution containing 9 ml of sterile broth. This step results in a dilution factor of 10, or 1:10, compared to the original culture. From this culture, another 1 ml is taken and mixed with a new tube of 9 ml of dilution solution, creating a dilution factor of 1:100 related to the original culture. This process continues until a series of dilutions is created that will bracket the desired cell concentration for accurate counting. Samples from each tube are plated onto the solid medium using either the pour plate or spread plate method, and the plates are incubated until colonies appear. Generally, 2 to 3 plates are prepared from each dilution, and the number of colonies on each plate is averaged. It is essential to mix these samples thoroughly with the dilution medium to ensure random cell distribution in the tube, which is critical for obtaining reliable results. The dilution factor is utilized to calculate the number of cells in the original cell culture. In the provided example, an average of 50 colonies was counted on the plates from 1:10,000 dilution. Since only 0.1 ml of suspension was pipetted into the plate, the multiplier needed to restore the original concentration is 10 multiplied by 10,000. Therefore, the number of CFU per ml is calculated as 50 multiplied by 10 multiplied by 10,000 equal to 5 million. This indicates that the estimated number of bacteria in the culture is 5 million cells per ml. The colony count from the 1:1000 dilution was 389, which is significantly below the expected count of 500 for a tenfold difference in dilutions. This discrepancy underscored the potential inaccuracy when colony counts exceed 300, as multiple bacterial cells may grow into a single colony. The number of microorganisms in dilute sample is often too low to be detected by the plate count method described so far. For these specimens, microbiologists typically use the most probable number or MPN method, a statistical procedure used for estimating the number of viable microorganisms in a sample. This method is commonly applied to the water and food samples, evaluating detectable growth by observing changes in turbidity or color resulting from metabolic activity. A typical application of the MPN method is estimating the number of coliforms in a sample of pond water. Coliforms are gram-negative rod-shaped bacteria that ferment lactose. The presence of coliforms in water indicates potential contamination by fecal matter. In the method illustrated, a series of 3 dilutions of a water sample is tested by inoculating 5 lactose broth tubes with 10 ml of sample, 5 lactose broth tubes with 1 ml of sample, and 5 lactose broth tubes with 0.1 ml of sample. The lactose broth tubes contain a pH indicator that changes in color from red to yellow when lactose is fermented. After inoculation and incubation, the tubes are examined for signs of coliform growth by a color change in the media from red to yellow. In the first set of tubes, 10 ml sample, growth is observed in all the tubes. In the second set, 1 ml, growth is seen in only 2 out of the 5 tubes. In the third set, 0.1 ml, no growth is observed in any of the tubes. The number 5, 2, and 0 are compared with a reference figure that has been created using probability model of sampling procedures. From this comparison, we conclude that 49 is the most probable number of bacteria per 100 ml of sample. In addition to direct methods of counting cells, indirect methods are frequently employed to estimate and compare cell densities in cultures. One primary approach is to measure the turbidity or cloudiness of a bacterial sample in a liquid suspension. The instrument utilized for measuring turbidity is known as a spectrophotometer. In a spectrophotometer, a beam of light is transmitted through the bacterial suspension. A detector measures the light that passes through the suspension, converting the amount of light reaching the detector into either percent transmission or a logarithmic value called absorbance or optical density. As the number of bacteria in a suspension increases, the turbidity also rises, resulting in a less light reaching the detector. This decrease in light that passes through the sample correlates with a decrease in percent of transmission and an increase in absorbance measured by the spectrophotometer. Measuring turbidity is a rapid method for estimating cell density, provided there are enough cells in a sample to create turbidity. It is possible to correlate turbidity readings with an actual number of cells by conducting a viable plate count of samples taken from cultures with a range of absorbance values. Using these values, a calibration curve can be generated by plotting turbidity against cell density. Once the calibration curve is established, it can be used to estimate cell counts for all samples obtained or cultured under similar conditions, with densities within the range of values used to create the curve. Binary fission is the most common pattern of cell division in prokaryotes, but it is not the only one. Other mechanisms usually involve asymmetrical division, as in budding, or the production of spores in aerial filaments. In some cyanobacteria, many nucleoids may accumulate in an enlarged round cells or along a filament, leading to the generation of many new cells at once. The new cells often split from the parent filaments and float away in a process called fragmentation. Fragmentation is commonly observed in the Actinomycetes, a group of gram-positive anaerobic bacteria commonly found in soil. Another curious example of cell division in prokaryotes reminiscent of live birth in animals is exhibited by the giant bacterium Epulopiscium. Several daughter cells grow fully in the parent cells, which eventually disintegrate, releasing the new cells in the environment. Other species may form long, narrow extension at one pole in a process called budding. The tip of the extension swells and forms a smaller cell, the bud, that eventually detaches from the parent cell. Budding is common in yeast, and it is also observed in prosticate bacteria and some cyanobacteria. This slide, a few more examples of alternative patterns of cell divisions are shown. An electron micrograph shows a budding Listeria monocytogenes cells in the A figure. The illustration B shows the cell divided into multiple fission in Cyanobacterium dermocarpa. The C is a micrograph, and an illustration shows spore, germ tube, vegetative hypha, branch, and aerial hypha in the Streptomyces spore formation. Microorganisms primarily grow in biofilms, which are complex ecosystems that form on various surfaces, including industrial pipes, water treatment systems, natural environments like river rocks. These biofilms can develop almost on any surface in liquid environments with minimal nutrients. For instance, microbial mats that float on water consist of many photosynthetic microorganisms, while biofilms in the human mouth can harbor hundreds of bacterial species. Importantly, biofilms are not just a random cluster of microorganisms, they are structured communities that offer selective advantages to the microorganisms within them. Emergent properties of biofilm. Properties could not be predicted from studying single cell. Physiological changes and the EPS, extracellular polymeric substances, protect microbes from harmful agents, that is ultraviolet. When formed on medical devices such as implants, antibiotic treatment fails. Chunks of biofilm can be sloughed off, which can contaminate a drinking water system. Structure. Observation using confocal microscopy have demonstrated that environmental conditions significantly influence the structure of biofilms. In rapidly flowing water such as freshwater streams and specially designed laboratory flow cells, filamentous biofilms known as streamers form. These streamers are anchored to the substrate by a head, while their tail floats downstream with the current. In contrast, biofilms in still or slow-moving water typically take a mushroom-like shape. Additionally, factors like nutrient availability can cause further changes in biofilm structure. Detailed studies utilizing confocal laser scanning microscopy have revealed that biofilm consists of clusters of microorganisms embedded in an extracellular matrix, interspersed with open water channels. This extracellular matrix is primarily composed of extracellular polymeric substances, or EPS, secreted by the organisms within the biofilm. The EPS is a significant component of the biofilm, making up to 50 to 90% of its total dry mass. And its properties can vary based on the types of organisms present and environmental conditions they experience. EPS is a hydrated gel predominantly made by polysaccharides along with other macromolecules such as proteins, nucleic acids, and lipids. It plays a crucial role in maintaining the biofilm's integrity and function by facilitating movement of nutrients, waste, gaseous throughout the structure. The dynamic system helps keep the cells hydrated, thereby preventing desiccation. Moreover, EPS provides protection to the organism in the biofilm from predation by other microbes or cells such as protozoans and human white blood cells. Biofilm formation begins when free-floating microbial cells known as planktonic cells attach to a surface coated with a conditioning film of organic material. This attachment occurs in multiple stages, starting with reversible attachment. As the cells adapt and produce extracellular polymeric substances, they transition to a sessile lifestyle, forming structured biofilm that includes an extensive matrix and water channels. Specialized appendages such as fimbriae, pili, and flagella play a crucial role in this process, aiding the establishment of a mature biofilm. Eventually, cells at the biofilm periphery can revert to a planktonic stage, detaching and dispersing to colonize a new environment. In a biofilm, diverse microbial species engage in metabolic interactions, where the waste of one organism serves as a nutrient for another. For instance, aerobic microorganisms consumes available oxygen, leading to the creation of an anaerobic zone that nurture anaerobic species. This dynamic exchange is commonly observed in polymicrobial infections where both aerobic and anaerobic pathogens coexist and collaborate, highlighting the complexity and interdependence of microbial communities within the biofilm. Biofilms and human health. The human body is home to various biofilms, some of which are beneficial while others can be harmful. Beneficial biofilms such as normal microbiota found in intestinal and respiratory mucosa help protect against infections from pathogens. Conversely, harmful biofilms like dental plaque can lead to dental and periodontal diseases. Additionally, biofilms can develop in wounds, leading to serious infections, and are particularly problematic in patients with cystic fibrosis, where the bacterium Pseudomonas aeruginosa often colonizes the airways, causing chronic and potentially fatal lung infection. Biofilms can also form on medical devices, resulting in infections in individuals with indwelling catheters, artificial joints, or contact lenses. Biofilms present a significant challenge in treating infections due to their increased resistance to antibiotics compared to free-floating bacteria. Several theories explain this enhanced resistance, including the metabolic inactivity of the cells located deeper into the biofilm, which makes them less vulnerable to antibiotics and target metabolic processes. The extracellular polymeric substances, or EPS, can hinder the diffusion of antibiotics, preventing effective treatment. Additionally, phenotypic changes such as a heightened production of efflux pump and that expels antibiotic from the bacterial cells contribute to resistance. Biofilms also facilitate the exchange of extrachromosomal DNA, which includes genes that confer antibiotic resistance, further complicating treatment efforts. Quorum sensing is a mechanism that allows in a biofilm to coordinate their activities in response to environmental stimuli. This process enables microorganisms to detect their cell density through the release and binding of a small diffusible molecule known as autoinducers. When the population reaches a critical threshold, these autoinducers trigger a series of reactions that activate genes important for functions that are advantageous only at high cell densities. For instance, certain pathogens begin to produce virulence factors only when there are cells present to successfully evade the host immune system. Quorum sensing not only occurs among bacterial population, but also between bacteria and eukaryotes, including interactions with human-associated fungi like Candida albicans, which can lead to infections in individuals with weakened immune systems. The signaling molecules involved in quorum sensing are classified into two main types. Gram-negative bacteria primarily communicate with N-acylated homoserine lactones, while gram-positive bacteria mainly utilize small peptides. The initial step in quorum sensing involves the binding of an autoinducer to its specific receptor once a sufficient concentration of signaling molecule is present. This binding initiates a cascade of signaling events that ultimately alters gene expression. Consequently, biological responses linked to quorum sensing are activated, particularly resulting in an increase in the production of signaling molecules themselves, reinforcing the concept of autoinduction. Quorum sensing is a cell-to-cell communication process that allows bacteria to coordinate their behavior based on population density. This phenomenon is particularly evident in the relationship between the bobtail squid and the bioluminescent bacteria Vibrio fischeri. The bobtail squid possesses a specialized light organ that houses Vibrio fischeri. Through quorum sensing, Vibrio fischeri can detect its own population density, and in response, it regulates the expression of the bioluminescence genes. When the bacterial population reaches a critical threshold, they emit light, which helps the squid blend into the moonlight, effectively camouflaging it from the predators. The mutualistic relationship is beneficial for both species. The squid gains protection while providing nutrients for the bacteria. Furthermore, the Vibrio fischeri has garnered interest in fisheries due to its role in marine ecosystems. Understanding quorum sensing mechanisms can enhance aquaculture practices by promoting beneficial microbial communities. By harnessing the natural interaction between bacterium and marine organisms, researchers aim to improve the fish health and optimize production in fisheries, showcasing the importance of microbial ecology in sustainable practices. The bobtail squid and the Vibrio fischeri exemplify the intricate connection within marine environments, highlighting the significance of microbial communication. The essential requirements for life are often thought to include water and oxygen, but the role of oxygen is more complex than commonly assumed. While water is universally acknowledged as vital, molecular oxygen was not always present on Earth. The earliest life forms emerged in an environment devoid of free oxygen. It was only after the cyanobacteria began to release oxygen through photosynthesis that atmospheric oxygen levels rose. This increase, known as a Great Oxygenation Event or Oxygen Revolution, led to significant extinction event as many organisms could not withstand the damaging effect of the reactive oxygen species or ROS generated from oxygen. Reactive oxygen species include singlet oxygen, which is excited state of the molecular oxygen with paired electron spins, making it more reactive than triplet oxygen, which is in the ground state of oxygen with two unpaired electrons with parallel spins. Essentially, singlet oxygen has a lower spin multiplicity, which is singlet, compared to the triplet oxygen's higher spin multiplicity, or triplet. These differences make singlet oxygen highly reactive. In addition, ROS also includes highly unstable and reactive superoxide and peroxides, hydroxyl radicals, and hypochlorite ions, all of which can harm biological macromolecules. However, some organisms adapted to detoxify these reactive species by utilizing the electronegativity of oxygen to generate free energy for their metabolism. These adaptations allowed certain life forms to thrive in the newly oxygen-rich environment, demonstrating that while oxygen is not always necessary for life, its presence can significantly influence the evolution and survival of organisms. Many ecosystems are still free of molecular oxygen. Some are found in extreme locations, such as deep in the oceans or in Earth's crust. Others are part of our everyday landscape, such as marshes, bogs, and sewers. Within the bodies of humans and other animals, regions with little or no oxygen provide an anaerobic environment for microorganisms. We can observe different requirements for molecular oxygen by growing bacteria in thioglycolate broth tube cultures. A test tube culture begins with an autoclaved thioglycolate broth medium, although sometimes a low percentage of agar can be used for free bacterial movements throughout the tube. Thioglycolate possesses strong reducing properties, and autoclaving removes most of the oxygen. The tubes are inoculated with bacterial cultures to be tested and incubated at an appropriate temperature. Over time, oxygen slowly diffuses from the top of the thioglycolate tube cultures. Bacterial density increases in the areas where the oxygen concentration is most suitable for the growth of that particular organisms. The growth of bacteria with varying oxygen requirements in the thioglycolate tubes is illustrated in the slide figure. In the first tube A, growth is concentrated at the top. The bacteria in the tube are obligate or strict aerobes that cannot grow without an abundant supply of oxygen. Conversely, the tube B exhibits heavy growth at the top and throughout the tube, a typical result with facultative anaerobes. Facultative anaerobes thrive in the presence of oxygen but can also grow in its absence of relying on fermentation and anaerobic respiration, provided there is a suitable electron acceptor other than oxygen. The tube C shows growth at the bottom. These bacteria are obligate anaerobes which are harmed by oxygen. Aerotolerant anaerobes in tube D are indifferent to the presence of oxygen. They do not utilize oxygen because they typically have a fermentive metabolism, but they are not harmed by the oxygen like obligate anaerobes are. Tube E shows a Goldilocks culture where the oxygen level must be just right for the growth, neither too much nor too little. These microaerophiles require a minimum level of oxygen for growth, approximately between 1% and 10%, which is well below the 21% found in the atmosphere. Examples of obligate aerobes include Mycobacterium tuberculosis, the causative agent of tuberculosis, and Micrococcus luteus, a gram-positive bacterium that colonizes the skin. Neisseria meningitidis, responsible for severe bacterial meningitis, Neisseria gonorrhoeae, which causes sexually transmitted gonorrhea, are also obligate aerobes. Many obligate anaerobes thrive in an environment where anaerobic conditions exist, such as deep sediments of soil, still waters, and bottom of the deep ocean where there is no photosynthetic life. Anaerobic conditions also occur naturally in the intestinal tract of animals. Obligate anaerobes, primarily the bacteroids, make up the significant fraction of the microbes in the human gut. Transient anaerobic conditions arise when the tissue are deprived of blood circulation, creating an ideal breeding ground of obligate anaerobes. One type of obligate anaerobe found in the human body is the gram-positive rod-shaped Clostridium species. Their ability to form endospores enables them to survive in the presence of oxygen. Clostridium difficile, commonly known as C. diff, is a major cause of health-acquired infections. Prolonged antibiotic use and other infections increase the risk of developing secondary Clostridium difficile infection by disrupting the balance of microorganisms in the intestine, allowing C. difficile to colonize and causing significant inflammation of the colon. Other Clostridia responsible for serious infections include Clostridium tetani, the agent of tetanus, and Clostridium perfringens, which causes gas gangrene. In both instances, the infection originates in necrotic tissues, which are dead tissues lacking oxygen from blood circulation. This explains why deep puncture wounds are associated with tetanus. When tissue death occurs alongside a lack of circulation, gangrene remains a constant danger. Staphylococci and Enterobacteriaceae are the types of facultative anaerobes. Staphylococci are commonly found on the skin and in the upper respiratory tract, while Enterobacteriaceae primarily reside in the gut and the upper respiratory tract. However, they can occasionally spread to the urinary tract, leading to infections. it is not uncommon to observe mixed bacterial infections where facultative anaerobes consume oxygen, creating an environment that allows an obligate anaerobes to thrive. Examples of aerotolerant anaerobes include lactobacilli and streptococci, both present in the oral microbiota. An example of microaerophile is Campylobacter jejuni, which is responsible for gastrointestinal infection and grows best in low oxygen concentration. The optimum oxygen concentration, as the term suggests, refers to the ideal level of oxygen for specific microorganisms. The minimum permissive oxygen concentration is the lowest level of oxygen that supports growth. Conversely, the maximum permissive oxygen concentration is the highest level of tolerated by an organism. Growth will not occur outside the range defined by these minimum and maximum permissive oxygen concentrations. Capnophiles are bacteria that thrive in environments with higher concentration of carbon dioxide and a lower concentration of oxygen than those found in the atmosphere. A common method to cultivate these bacteria involves using a candle jar, which is a jar with a tight-fitting lid designed to hold the cultures, and a candle. Once the culture are placed inside, the candle is lit, the lid is closed, allowing the burning candle to consume most of the oxygen while releasing CO2, creating an ideal environment for the growth of capnophiles. The study of obligate anaerobes requires special equipment, as these bacteria must be grown in conditions devoid of oxygen. The most common method is culturing in an anaerobic jar, which includes a chemical packs that remove oxygen and release carbon dioxide. Alternatively, an anaerobic chamber is an enclosed box from which all oxygen is removed. Gloves sealed to openings in the box allow for handling of the culture without exposing them to air. Aerobic respiration constantly generates reactive oxygen species byproducts that must be detoxified. Even organisms that do not use aerobic respiration need some way to break down some of the ROS that may form from atmospheric oxygen. Three main enzymes break down these toxic byproducts: superoxide dismutase or SOD, peroxidase, and catalase. Each one catalyzes a different reaction. Superoxide dismutase break down powerful superoxide anions generated by aerobic metabolism into harmless molecular oxygen and harmful hydrogen peroxide. Hydrogen peroxide can be further detoxified either of the two or both enzyme, peroxidase or catalase. Peroxidase. In this reaction, an electron donor reduced compound, example reduced nicotinamide adenine dinucleotide or NADH, oxidizes hydrogen peroxides or other peroxides to water. The enzyme play an important role by limiting the damage caused by peroxidation of membrane lipids. Catalase. The enzyme catalase converts hydrogen peroxide to water and molecular oxygen. On the basis of this oxygen formation, a catalase test can be done. See the inset in the slide figure. Obligate anaerobes usually lack all three enzymes. Aerotolerant anaerobes do have SOD but no catalase. On the basis of the catalase test, microbiologists can distinguish streptococci, which are aerotolerant and do not possess catalase, from staphylococci, which are facultative anaerobes. The learning objective includes illustrating and describing the minimum, optimum, and maximum pH requirements for microbial growth, identifying the categories of microbe based on their pH needs, namely acidophiles, neutrophiles, and alkaliphiles, and providing examples of microorganisms for each pH category. Yogurt, pickles, sauerkraut, and lime-seasoned dishes have a tangy taste due to their high acid content. Acidity is determined by the concentration of hydrogen ions and measured by using pH. Environments with pH values below 7 are acidic, while those above 7 are basic. Extreme pH levels can affect the structure of all macromolecules. For instance, high pH can break the hydrogen bonds in DNA, and extremely basic conditions can hydrolyze lipids. The protonomotive force essential for ATP production in cellular respiration relies on the concentration gradient of H+ across the plasma membrane. If the protons are neutralized by hydroxide ions, this gradient collapses, impairing energy production. Proteins, the cell's workhorses, are particularly sensitive to pH changes. Even moderate shifts in pH can alter ionization of amino acid functional groups and disrupt hydrogen bonding, leading to changes in the protein's folding, denaturation, and loss of function. The optimum growth pH refers to the most favorable pH for an organism's growth, while the minimum and maximum growth pH values indicate the extremes of the pH tolerance for that organism. These pH values vary significantly among different organisms, which is crucial for the food preservation and survival of microorganisms in various environments, such as human stomach. For instance, Salmonella species has an optimum growth pH of 7 to 7.5, with a minimum growth pH around 4.2. Most bacteria, including common species like Escherichia coli and Staphylococci, are classified as neutrophiles, thriving at pH levels close to neutral. However, certain pathogenic strains show greater resilience to acidic conditions, unlike most familiar bacteria. Fungi, in contrast, tend to flourish in a slightly acidic conditions, with an optimal pH range of 5 to 6. Microorganisms that thrive at pH levels below 5.55 are termed acidophiles. For example, Sulfolobus species, which are sulfur-oxidizing archaea, found in harsh environments like Yellowstone National Park, can survive in extremely acidic conditions with pH values between 2.5 and 3.5. Additionally, Ferroplasma species can exist in even more extreme acidic environments with pH levels ranging from 0 to 2.9. Lactobacillus bacteria, which are the part of the normal vaginal microbiota, can endure acidic conditions with a pH value ranging from 3.5 to 6.8, contributing to the vagina's acidity by producing lactic acid. Acidophilic microorganisms possess unique adaptations that enable them to thrive in highly acidic environments. These adaptations include an increased negative surface charge on proteins that enhances their stability at low pH levels, as well as specialized pumps that actively remove protons from their cells. Additionally, alterations in the composition of the membrane phospholipids help maintain membrane fluidity in acidic conditions. The acidity of the vagina, which generally has a pH of around 4, plays a critical role in inhibiting growth of less acid-tolerant microbes, thereby supporting the health of microbial ecosystem in that area. The alkaliphiles are microorganisms that thrive at pH levels between 8 and 10.5, with Vibrio cholera growing optimally at pH 8, though it can withstand up to pH 11 but is rendered inactive by stomach acid. The archaean Natronobacterium found in Africa's soda lakes can survive at pH of 10.5. These organisms have evolved adaptations in their lipid and protein structure to survive high pH conditions, with Bacillus firmus utilizing a sodium-ion gradient for energy instead of a proton motive force. Additionally, enzymes from alkaliphiles possesses a higher isoelectric point due to an increased number of basic amino acids compared to those from the neutrophiles. The learning objective focuses on understanding the temperature requirements for microbial growth by describing minimum, optimum, and maximum temperature needed. Additionally, it aims to identify the categories of different types of microbes based on their temperature preferences, including psychrophiles, psychotrophs, mesophiles, thermophiles, and hyperthermophiles, while providing examples of microorganisms within each category. Microbes can be classified based on their temperature ranges at which they can thrive. For instance, the core temperature of the human body is approximately 37 degrees Celsius, which aligns with the growth preference of many normal human microbiota and pathogens, such as E. coli, Salmonella, Lactobacillus species. These organisms are categorized as mesophiles, thriving in moderate temperature environments. On the cooler end of the temperature spectrum, organisms known as psychotrophs or psychotolerant microbes prefer temperatures ranging from a high of 25 degrees centigrade down to refrigeration temperature around 4 degrees. They are commonly found in temperate climate and play a significant role in the spoilage of refrigerated foods. Organisms that thrive in cold environments, such as those retrieved from Arctic lakes like Lake Williams, are classified as extreme psychrophiles. Psychrophiles can grow at 0 degrees Celsius and below with optimal growth near 15 degrees Celsius and generally do not survive above 20 degrees Celsius. Mesophiles are adapted to moderate temperature with optimal growth occurring at their specific temperature ranges. The highest growth rates for these organisms are found at their optimum growth temperature. Each microorganism has a minimum growth temperature for survival and replication as well as a maximum growth temperature. These ranges can vary based on the other environmental factors. Thermophiles or heat-loving organisms grow optimally in temperatures between 50 degrees Celsius and 80 degrees and do not multiply at room temperature. They are found in hot springs, geothermal soils, man-made environments such as compost piles, where they help decompose organic material. Notable examples of thermophiles include Thermus aquaticus and Geobacillus species. At the extreme end of temperature scale, hyperthermophiles exhibit growth at temperature ranging from 80 degrees to maximum of 110 degrees Celsius, with some surviving temperatures above 121 degrees Celsius, which is atypical for autoclaving. Hydrothermal vents in the ocean exemplify extreme environment with temperatures reaching around 340 degrees Celsius. Microbes isolated from these vents, such as Pyrolobus and Pyrodictyum, demonstrate optimal growth above 100 degrees Celsius and can survive autoclaving conditions. The growth pattern of these microorganisms are represented in a skewed curve but illustrate their temperature-dependent growth characteristics. Antarctica, known as the coldest place on Earth, is the home of surprising array of microscopic life. Recent research has revealed beneath its thick ice sheet lies a rich microbial ecosystem with organisms thriving in environments devoid of sunlight for millions of years. This discovery emphasizes the resilience and adaptability of life in extreme conditions. A significant finding was made in Lake Williams, located beneath about 2,625 feet of ice in West Antarctica. Researchers reported the presence of nearly 4,000 species of microbes in this subglacial lake, marking it the first time organisms have been retrieved from such a location. Brent Christner, a microbiologist, a lead author of the study, expressed excitement about the discovery, highlighting that it showcases an active ecosystem rather than just a presence of life. Antarctica contains approximately 400 lakes trapped under its ice sheet, with some, like Lake Williams, interconnected by rivers and streams. The implications of this discovery extend to other hidden waterways, potentially indicating that they may also harbor life. Martin Tranter, a glaciologist, described the study as a landmark in polar sciences, likely inspiring further research into subglacial lakes. The breakthrough in accessing Lake Williams occurred in January 2013 after extensive planning and significant investment by the National Science Foundation. The Wizard team utilized a custom hot water drill equipped with a decontamination system to extract water from the lake. Tests conducted shortly after retrieving the water confirmed the presence of life, revealing a vibrant ecosystem within. Researchers collected 8 gallons of lake water and sediment samples, later analyzing them at various institutions. The results indicated a surprising diversity, with 3,931 species of a single-cell organisms identified. Krista noted that the number of organisms found in Lake Williams is comparable to that of a surface lakes, challenging previous assumptions that about life in such isolated conditions. The microorganisms in Lake Williams survive without sunlight, relying on minerals and lake sediments for energy to fix carbon dioxide and organic compounds. The most prevalent microbe is an archaea that oxidizes ammonium, which upon dying serves as the substance for other microbial groups that utilize sulfur and iron for energy. Bacteria that consumes methane also contribute to the ecosystem complexity. The environment of the Lake Williams is unique, resembling the deep ocean floor rather than a typical pond or lake. Its muddy coloration results from the glacial floor, a fine sediment produced by glacial processes. The lake is relatively shallow, only about 6.5 feet deep, and spans about 23 square miles, situated within an ice pocket shaped by the pressure of the ice above. The water temperature hovers slightly below freezing, highlighting the harsh condition in which these microbes thrive. Lake Williams is a part of a dynamic system, with its water filling and draining in a cycle akin to a bathtub over a period of 5 to 10 years. Historical geological events indicate that the sea once flooded the region now occupied by the lake, contributing to the presence of ammonium and methane from ancient marine sediments. Krishner, a professor at the Louisiana State University, likened this unique area to southern Louisiana, albeit with a kilometer of ice overhead, illustrating the extraordinary nature of life beneath Antarctic ice. Microorganisms need water to grow as well as nutritional solutes in them. When solutes dissolve in water, they interact with water molecules through forces like hydrogen bonding, effectively tying up some of the water molecules and making them less free to participate in other interactions. The amount of moisture available is measured as water activity, or AW, which is the ratio of the vapor pressure of the substance being measured to the vapor pressure of the pure distilled water. Thus, AW of water is equal to 1. Bacteria require a high AW 0.97 to 0.99, while fungi can survive drier conditions. For example, the growth range of Aspergillus species is between 0.8 and 0.75. Osmotolerant microorganisms can grow over a wide range of water activity, but optimally at higher levels, while zero-tolerant microbes can withstand high solute concentrations. The relationship between the water activity and microbial growth is significant in food preservation and safety. When the water activity is reduced, either through drying, salting, or addition of sugar, microbial growth is inhibited, extending the shelf life of food products. This principle is utilized in various food preservation methods such as dehydrating fruits, curing meats, making jam and jellies. Understanding the impact of water activity on microbial growth is essential for food scientists and producers to ensure food safety and quality. For instance, low water activity environments can create conditions that favor the growth of specific molds, which can produce mycotoxins harmful to human health. Conversely, high water activity can produce the growth of pathogenic bacteria, posing a risk of foodborne illnesses. Therefore, monitoring and controlling water activity in food products is vital. Using water activity measurements, producers can make informed decisions about processing, packaging, and storing food products to prevent spoilage and maintain safety. In conclusion, water activity is a key determinant of microbial growth, influencing food preservation strategies and ensuring public health safety. Most natural environments usually have lower solute concentration compared to cytoplasm of many microorganisms. Rigid cell walls help protect cells from bursting in dilute environments. However, there is not much protection against high osmotic pressure. In such cases, water flows out of the cell following its concentration gradient. This leads to plasmolysis, the shrinking of the protoplasm away from the intact cell walls, and ultimately cell death. This phenomenon explains why using brines and layering meat and fish in salt are traditional methods of food preservation. Microorganisms known as halophiles, salt-loving, actually require high salt concentration for their growth. These organisms thrive in marine environments where salt concentrations are around 3.5%. Extreme halophilic microorganisms such as red alga, Dunaliella salina, and an archaeal species Halobacterium grow in hypersaline lakes, like in the Great Salt Lake, which is 3.5 to 8 times saltier than the ocean, and the Dead Sea, which is 10 times saltier than the ocean. Dunaliella species encounters the immense osmotic pressure of their environment by maintaining a high cytoplasmic concentration of glycerol and actively pumping out salt ions. Halobacterium species accumulates large concentration of potassium and other ions in its cytoplasm. Its proteins are specifically designed to function under high salt concentration and lose activity at salt concentration below than 1 to 2 molar. While most halotolerant organisms, such as Halomonas species found in salt marshes, do not require high salt concentration for growth, they can survive and reproduce in salty conditions. It is not surprising that Staphylococci, Micrococci, Corynebacteria, which inhabit our skin, can tolerate salt in their environment. Halotolerant pathogens pose significant risk for foodborne illnesses since they can survive and multiply in salty foods. For instance, the halotolerant bacteria Staphylococcus aureus, Bacillus cereus, Vibrio cholerae produce harmful enterotoxins that are major contributors to food poisoning. Lake Natron and its surrounding in the northern Arusha region of Tanzania. The lake is mostly inhospitable to life except for a few species adapted to its warm, salty, and alkaline water. The deepest water along the perimeter of the lake is visible. The natural color image was captured on 3rd March 2017, very early in the rainy season, by the operational land imager OLI, the NASA's Landsat 8 satellite. The lake is a maximum of 35 miles long and 14 miles wide and shallow, less than 10 feet deep. The surrounding area receives irregular seasonal rainfall above 30 inches, mainly between December and May. Temperatures at the lake are frequently above 40 degrees Celsius to up to 60 degrees Celsius. High levels of evaporation have left behind natron, sodium carbonate decahydrate, and trona, sodium sesquicarbonate dihydrate. The alkalinity of the lake can reach to pH greater than 12. The surrounding bedrock is composed of alkaline, sodium-dominated trachyte lavas that were laid down during the Pleistocene period. The lavas have significant amounts of carbonate but very low calcium and magnesium levels. This has allowed the lake to concentrate into a caustic alkaline brine. The chemical properties of the water are known to calcify the bodies of any living thing that died in the lake. The color of the lake is characteristic of those where very high evaporation rates occur. As water evaporates during the dry season, salinity level increases to the point that salt-loving microorganisms begin to thrive, such as halophile organisms include some photosynthesizing cyanobacteria. The red accessory photosynthesizing pigments in the cyanobacteria produces the deep red of the open water of the lake and the orange color of the shallow parts of the lake. The alkali salt crust on the surface of the lake is also often colored red or pink by the halophilic microorganisms that live there. Salt marshes and freshwater wetlands around the edges of the lake do support a variety of plants. Lake Natron serves as the sole regular breeding area in East Africa for 2.5 million lesser flamingos, which are classified as near threatened due to their reliance on this specific habitat. The lake's increasing salinity encourages the growth of halophilic cyanobacteria, supporting the flamingo's nesting needs. These birds, forming the largest flock in East Africa, feed on spirulina, a blue-green algae rich in red pigments found in nearby saline lakes. The caustic conditions of the Lake Natron provides a protective barrier against predators, allowing the flamingos to nest safely on seasonally created evaporate islands, while greater flamingos also utilize the mudflats for breeding. The unique characteristics of Lake Natron have inspired the nature documentary The Crimson Wing: Mystery of the Flamingos by Disney Nature, highlighting the deep connection between this lake and the lesser flamingos, showcasing it as the only consistent breeding ground. Microbial life near underwater hydrothermal vents presents a captivating area of research due to distinctive ecosystems that emerge in these extreme environments. hydrothermal vents located along the mid-ocean ridges release pressurized superheated 340 degrees Celsius, which is 700 degrees Fahrenheit, mineral-rich water from the Earth's crust, creating a habitat that is both harsh and vibrant. Microbes found near hydrothermal vents include archaea, bacteria, and microbial mats, playing a crucial role in these unique ecosystems. These microbes are chemoautotrophic, meaning they produce their own food using chemicals present in the hydrothermal fluids. Among the archaea, Methanospirus canderi thrives on hydrogen gas, generates methane, while Pyrolobus also utilizes hydrogen gas but produces hydrogen sulfide. Bacteria, particularly chemolithotrophs, are the most plentiful and rely on sulfur as their energy source. Some, referred to as symbiotic vent bacteria, inhabit the tissues of vent organisms like tube worms and mussels. Notable genera of bacteria in these vents include Marinobacter, Vibrio, Pseudoalteromonas, Halomonas, Pseudomonas, and Alcanivorax, which are generalist species. Additionally, microbial mats, which can be iron oxide or manganese oxide encrusted, are rapidly formed structures that can reach several centimeters in depth. One of the most notable inhabitants of these ecosystems is the giant tube worm, known scientifically as Riftia pachyptila. These extraordinary creatures can grow up to 3 meters in length and lack traditional mouth or digestive systems. They depend on symbiotic relationships with chemosynthetic bacteria that resides within their bodies, specifically in the specialized tissues called trophosomes. The partnership is mutually beneficial. The tube worms provide a safe habitat and necessary compounds for the bacteria, while the bacteria supply the worms with essential organic molecules such as sugars and amino acids. A piezophile, older term barophile, is an organism that thrives under high hydrostatic pressure, specifically at pressures of 10 megapascal or more. The term originates from the Greek word piezo, meaning pressure, and phile, meaning loving. This classification was replaced the earlier term from barophile, as baro relates to weight rather than the pressure. In the context of extremophiles, piezophiles exist in environments where humans would consider hydrostatic pressures extreme, while these conditions are normal for the organism. A further classification, hyperpiezophiles, refer to those that grow optimally at presence exceeding 50 megapascal. Despite the harmful effects of high pressure on organisms that typically grow at atmospheric levels, piezophiles require these conditions for optimal growth. They can often continue to grow significantly higher pressure, even above 100 megapascal, in their deep sea habitats. These adaptations allow them to thrive in environments that would be detrimental to other life forms. Their growth patterns illustrate how these organisms have evolved to utilize high-pressure conditions to their advantage. The first identified obligate piezophile was the psychrophilic bacterium Colwellia marinimini strain M41, isolated from Challenger Deep of Mariana Trench east of Philippines in the Pacific Ocean. Schwannella benthica strain DB21MT2, shown in the figure, is also another hyperpiezophiles collected at a depth of 10,898 meters of the Mariana Trench. Other notable hyperpiezophiles include thermophilic archaeon Pyrococcus ioannonensis strain CH1, found at a deep-sea hydrothermal vent. Both strains exhibit optimal growth pressures beyond 20 megapascal and can survive at pressure exceeding 100 megapascal. The highest recorded growth pressure is 140 megapascal for Colwellia marinimini MTCD1. It is important to distinguish between the obligate piezophiles, which cannot grow at lower pressures, and piezo-tolerant organisms, which can grow at lower rates under high pressure. It is extremely difficult to study these deep-sea piezophiles as they are uncultivable in lower pressure. Scientists have to isolate and keep the bacteria under its tolerated pressure all the time from the collection to the laboratory. Photoautotrophs such as cyanobacteria and green sulfur bacteria, along with photoheterotrophs like purple non-sulfur bacteria, require adequate light intensity at specific wavelengths absorbed by their pigments for growth and reproduction. These organisms capture energy from light through pigments, converting it into chemical energy that fuels carbon fixation and other metabolic activities. The range of electromagnetic spectrum they absorb is known as photosynthetically active radiation, which spans the visible light spectrum for 400 to 700 nanometers and extends into near-infrared for some photosynthetic bacteria. Accessory pigments including fucoxanthin in brown algae and phycobilanes in cyanobacteria expand the wavelength range usable for photosynthesis, helping to adapt to low light levels at greater water depths. Additionally, some microorganisms like Halobacteria archaea utilize light energy for their proton and sodium pumps, absorbing light through a pigment protein called bacteriorhodopsin, which is akin to the eye pigment rhodopsin. Photosynthetic bacteria thrive not only in aquatic environments, but also in soil and symbiotic relationship with fungi and lichens. A notable example is the microalga Chlamydomonas nivalis, which causes a phenomenon known as watermelon snow due to its richness in the secondary red carotenoid pigment astaxanthin that imparts a pink hue to the snow where it grows. The study of microorganisms is generally facilitated if we are able to culture them, that is, to keep reproducing populations alive under laboratory conditions. Culturing many microorganisms is challenging because of highly specific nutritional and environmental requirements and the diversity of these requirements among different species. The culture medium is a solid or liquid mixture of nutrients and other compounds essential for growth, transport, and storage of microorganisms in a laboratory setting. It must include all the necessary growth factors that a microorganism requires. When selecting a culture media, it is important to consider the microbe's normal habitat to ensure optimal growth conditions. Culture media can be classified based on their chemical compositions, physical nature, and their specific functions in supporting microbial life. A media for microbial culture can be chemically defined and mostly used in analytical laboratories procedure and analysis. While most of the growth cultures media in the laboratories and industry are complex media, the slide illustrates some, but not all, of the essential ingredients commonly found in some of the bacteriological media. The number of available media for growing bacteria is substantial. Some media are classified as general all-purpose media supporting the growth of a wide variety of organisms, with tryptic soy broth, TSB, being a prime example. Specialized media are utilized for bacterial identification and are often supplemented with dyes, pH indicators, or antibiotics. Enriched media, a specific type of specialized media, includes growth factors, vitamins, and other nutrients to promote the growth of fastidious organisms, which cannot synthesize certain nutrients and require them to be added. When the complete chemical composition of a medium is known, it is referred to as a chemically defined medium, such as an easy medium, where all individual chemical components and their extract amounts are identified. In contrast, complex media contains extract and digest from yeasts, meat, or plants, and their precise chemical composition is unknown, with individual components amounts being variable. Examples of complex media include the nutrient broth, tryptic soy broth, and brain hurt infusion. Media that inhibit the growth of unwanted microorganisms and support the growth of organisms of interest by supplying nutrients and reducing competition are called selective media. Crystal violet agar is exclusive selective media where only gram-negative organisms grow. The presence of crystal violet binding to thick cell wall peptidoglycan prevent the gram-positives to grow. Crystal violet acts as if their cell wall was glued in gram-positive, while due to the LPS layer, crystal violet can't penetrate the cell wall in gram-negatives, allowing them to grow. For example, if we want to isolate bacteria that break down crude oil, hydrocarbonoclastic bacteria, sequential subculturing in a media that supply carbon only in the form of crude oil will enrich the culture with oil-eating bacteria. The differential media make it easy to distinguish colonies of different bacteria by change in the color of colonies or the color of the media. Color changes are the result of end products created by the interaction of bacterial enzymes with their differential substrates in the medium, or in the case of hemolytic reactions, the lysis of the red blood cells in the media. The blood agar media is a classic differential media which can distinguish three types of bacteria exhibiting alpha, moderately hemolytic, partial hemolysis of red blood cells, beta, extreme hemolytic, total destruction hemolysis of the red blood cells, and non-hemolytic gamma, which is the least virulent types. The combination of the selective and differential media make it easy to select a certain type of bacteria as well as distinguish colonies of different bacteria by a change in the color of the colonies or the color of the media. Color changes are the result of end products created by interaction of bacterial enzymes with differential substrates in the medium, or in the case of hemolytic reaction, the lysis of the red blood cells in the media. Mannitol salt agar, or MSA, contains mannitol sugar, sodium chloride, high percentage 7.5 to 10%, phenol red indicator. They grow only select gram-positive bacteria, preferentially Staphylococcus, and inhibit gram-negative bacteria due to the high salt in the media, which is a selective property of the media. In addition, the presence of sugar mannitol allows gram-positive fermenters to ferment the sugar and produce organic acids, dropping the pH of the media. The presence of the phenol red allows gram-positive fermenters to be distinguished from non-fermenters. Phenol red is a water-soluble pH indicator dye that changes color from red to yellow in acidic pH. The fermenters can distinguish on the media by the presence of the changed yellow color around their growth. McConkey agar contains lactose sugar, bile salts, crystal violet, and neutral red indicator. McConkey agar contains lactose sugar, bile salts, crystal violet, and neutral red indicator. McConkey agar is a selective and differential culture medium primarily used for isolating and differentiating gram-negative bacteria, particularly member of the Enterobacteriaceae family. Most importantly, it contains lactose and a pH indicator neutral red. Lactose serves as a fermentable carbohydrate, allowing for the differentiation of lactose fermenters, which produce acid and turn the medium pink, from non-lactose fermenters, which do not produce acid, thus remain colorless. The selective components also include bile salt and crystal violet, which inhibit the growth of gram-positive bacteria, ensuring that only gram-negative bacteria can thrive on the medium. This unique combination allows for effectively identification and isolation of enteric bacteria in clinical and laboratory settings. Eosin methylene blue agar contains lactose sugar, eosin Y, and methylene blue dyes. EMB is a selective and differential culture medium used primarily for isolating identifying gram-negative bacteria, particularly those of the Enterobacteriaceae family as well. Eosin Y and methylene blue dye, which serves as both differential indicators and selective agents. These dyes inhibit the growth of gram-positive bacteria while allowing gram-negative bacteria to flourish. EMB is a selective and differential culture medium used primarily for isolating and identifying gram-negative bacteria, particularly those of the Enterobacteriaceae family. Eosin Y and methylene blue dyes, which serve as both differential indicators and selective agents. These dye inhibit the growth of gram-positive bacteria while allowing gram-negative bacteria to flourish. EMB agar specifically differentiates lactose fermenters from non-fermenters. The aggressive lactose fermenters such as E. coli produce a lot of organic acid which reacts with the dyes to form a dark metallic green sheen. The moderate fermenters produce a bubblegum pink coloration while non-fermenters remain colorless or take on the color of the media. This unique combination of components make EMB agar effective for distinguishing between various types of enteric bacteria in clinical and environmental microbiology. Clinical focus: Listeria monocytogenes. Jenny, a 24-year-old pregnant woman in her second trimester, visits a clinic with flu-like symptoms including a high fever, fatigue, and muscle aches. Despite her healthy lifestyle, which includes regular exercise and a nutritious diet, her healthcare provider decides to conduct a blood test due to concerns about her pregnancy, making her more susceptible to infection, especially Listeria monocytogenes. The blood samples are cultured on sheep blood agar, revealing small grayish colonies with clear zone indicating beta hemolysis. Additional tests confirm the presence of Listeria in her blood. This diagnosis suggests that Jenny has a listeriosis, a serious infection that poses 20% mortality risk to her fetus. Although a sample from her amniotic fluid tests negative for Listeria, a molecular test confirms that no bacteria crossed the placenta. Jenny is hospitalized and treated with high doses of antibiotics, including ampicillin or penicillin G, for 2 weeks. She fully recovers and released after a week. Listeria monocytogenes is a gram-positive bacteria found in various environments and is known to cause food poisoning, particularly in pregnant women due to their weakened immune system. It can cross the placenta, leading to severe complications. Pregnant women are advised to avoid certain foods, including soft cheeses and unpasteurized products, to reduce the infection risks. Prompt identification and notification of suspected listeriosis are crucial in the laboratory setting due to potential confusion with other gram-positive rods. It is possible to predict the number of cells in a population when they divide by binary fission at a constant rate. As an example, consider what happens if a single cell divides every 30 minutes for 24 hours. The diagram in the slide figure shows the increase in cell number for the first 3 generations. The number of cells increases exponentially and can be expressed as 2n, where n is the number of generation. If the cells divide every 30 minutes, after 24 hours, 48 divisions would have taken place. We apply this formula to n, where n is equal to 48, and the single cell would give rise to 2 to the power 48 cells at 48 generation, which is in 24 hours. When dealing with such huge numbers, it is more practical to use scientific notation. Therefore, we express the number of cells as 2.8 into 10 to the power 14 cells. In our example, we used 1 cell as the initial number of cells. For any number of starting cells, the formula is adapted as follows: that nn equal to n02 to the power n, where nn is the number of cells at any generation n. N0 is the initial number of cells, and n is the number of generations. Charles, a retired bus driver with type 2 diabetes, has become sedentary and gained weight since retiring. He experienced a tingling in his foot but dismissed it as normal. However, a non-healing scratch on his foot worsened, leading to a purplish discoloration and oozing. Concerned, he visited his physician and was quickly taken to a surgery for a diabetic foot ulcer. There was a risk of gas gangrene, likely caused by bacterium Clostridium perfringens, which thrives in oxygen-deprived dead tissue. Fortunately, the surgeon confirmed that the bone was not infected, and while the ulcer required surgical debridement, amputation was not necessary. Sadly, amputations are on the rise in the United States. About 160,000 people with diabetes undergo amputation each year. And 80% of non-traumatic lower limb amputations happen due to diabetes complications. An individual who has had amputations has worse chances of 5-year survival than someone with coronary artery disease, breast cancer, and colorectal cancer. These are some of the links to learning for educators resources. Thank you.